Good day to everybody. Today I am going to discuss about the effect of bacteriophage on starter culture. Actually, bacteriophage, we know that bacteriophage is one type of virus which will kill the bacterial cell. And starter culture, we know that starter culture are the carefully selected desirable microorganism. It may be bacteria, yeast and mold, which are deliberately added to milk or any other food product to initiate and accomplish the fermentation, desired fermentation. We know that in previous video I have discussed about this all these things in details. And fermentation is what? Fermentation, what is fermentation? Fermentation is the metabolic process in which energy release, with the release of energy. Fermentation is the metabolic process in which the carbohydrate and other related materials will be oxidized with the release of energy in absence of external electron acceptors, which is called fermentation. Now we know that. So today I am going to discuss about the different types of fermented milk products. First I will discuss about the different types of fermented milk products, and then I will discuss about the different types of fermented non-dairy fermented products. So first we are going to discuss about the fermented milk products. In the category of fermented milk products, most popular milk product is the dahi. We know that. Then yogurt will come. Then yakut. Then acidophilus milk. Then kefir, 6 kumis, which is also popularly known as milk wine because it contains some alcohol also, and teti, and eighth one is the sky. So these are the general milk products. Are fermented milk products are very popular in India and subcontinent, and also whole world, in whole world. Dahi, first of all, the dahi is the milk, is the base material for the dahi. Any tone milk or any skim milk can be used for the dahi production. And generally the starter culture, Lactococcus lactis, subspecies lactis, Lactococcus lactis, subspecies Chimoris, Streptococcus thermophilus, Lactobacillus bulgaricus, Lactobacillus plantarum. This type of spherical shape, cocci shape and rod shape bacteria or lactic acid bacteria are generally used for the production of doi. And second one is the yogurt. In yogurt, very interesting thing is happened because there are two types of one is cocci or spherical shape Streptococcus thermophilus, and another is the rod shape Lactobacillus delbrueckii subspecies bulgaricus. These two type of bacteria are used in proportion of 1:1, and they are living in the yogurt environment in a symbiotic relation, means they are helping each other. Like Lactobacillus bulgaricus are producing some amino acids like proline and glutamic acid, which will enhance the growth of Streptococcus thermophilus. Streptococcus thermophilus is also producing formate and carbon dioxide, which will promote the growth of Lactobacillus bulgaricus. So they are helping each other. In this way they are living in the in yogurt environment. And yakut, we know that yakut is a Japanese probiotic fermented milk product, and Lactobacillus casei strain Shirota is used for the production of the yogurt according to the name of the Professor Minoru Shirota, who discovered the this particular strain Lactobacillus casei strain Shirota. And acidophilus milk, as in acidophilus, the Lactobacillus acidophilus, the rod-shaped lactic acid bacteria are used for the fermentation of acidophilus milk. And kefir, in the kefir case, there are so many bacteria and yeast are present, like Lactobacillus brevis, Lactobacillus fermentum, Lactobacillus caucasus, and the Saccharomyces delbrueckii, Torulopsis, kefir, different types of bacteria and combination of bacteria and yeast are present. Also micrococci is also present. And interestingly, kumis is generally produced from mare's milk, not cow buffalo milk. It is produced from mare's milk, and it is also called, popularly known as milk wine, because it contains some amount of uh, alcohol also. So here also the Lactobacillus brevis, Lactobacillus bulgaricus, Saccharomyces lactis, Torulopsis, Torula cumis, and micrococci are generally used for the production of kumis. And teti is the Scandinavian ropey type milk, and it is generally bacteria and yeast are used for that. Bacteria like Lactococcus lactis, subspecies Hollandicus, and yeast like Saccharomyces Saccharomyces teti are generally used for the production of this fermented milk product. Teti, it is very popular in the Scandinavian countries like Denmark, Sweden, and Finland, like countries. And Sky is the traditional Finlandic fermented milk product. It is rich in protein and less in fat. And here the Streptococcus thermophilus, Lactobacillus bulgaricus, and Lactobacillus helveticus, this type of bacteria and lactic acid bacteria generally used for the production of Sky. Now we are going to discuss about this is the fermented milk products, dairy products. Now we are going to discuss about the fermented non-dairy food products. Fermented non-dairy food products. Fermented non-dairy food products. Among them, first one, we here come the bread. In bread also the alcoholic fermentation is happened in bread. In case of beer and wine, production of beer, there also the alcoholic fermentation will be happened. Like some of the sauerkraut, sorry, sauerkraut, sauerkraut, and pickles, also the fermented products. One by one I shall discuss about that. And also the miso, soya sauce, soya sauce, and the Indian traditional South Indian products like idli, dosa, dhokla. The dhokla is not a South Indian dairy particular South Indian dairy product. It is a North Indian dairy North Indian fermented food products. Sorry, they are not dairy products. Idli and dosa is the popular in South India also. So bread, first we come to the bread. Bread, the glucose is fermented by yeast Saccharomyces cerevisiae into alcohol and CO2. This is called the baker's yeast. Saccharomyces cerevisiae is called the baker's yeast. And the beer, in case of beer and wine, beer, you know that the malt product is will be fermented by the yeast. It is also alcoholic fermentation by yeast. Top fermenting is Saccharomyces cerevisiae, and bottom fermenting is Saccharomyces carlsbergensis. And in case of wine, the grapes juice are fermented by some yeast like Saccharomyces ellipsoides to produce the alcohol. You know that sorry cloth is here the raw material used for the sorry cloth is the cabbage, and here the fermenting bacteria are generally Leuconostoc mesenteroides and Lactobacillus plantarum. And in case of pickles, pickles is also fermented and raw material used for the pickle is generally cucumber. And the microflora used for the fermentation of pickles is the fermentation production of pickles is the Lactobacillus plantarum and Pediococcus cerevisiae. And miso is a popular Japanese soybean product. Here also the Aspergillus oryzae means the mold, and the Zygosaccharomyces roxy, the yeast, and the Lactobacillus delbrueckii bacteria are used for the production of miso. Soya sauce also the Aspergillus oryzae, Aspergillus oryzae, and the Zygosaccharomyces roxy and Lactobacillus delbrueckii are used for the production of soya sauce. And the temp is also the soybean Indonesian temp, TEMPEH, temp. It's also a very popular. Here raw material is the soybean, and here also the Rhizopus oryzae and Rhizopus oligosporas are generally this type of mold are used for the fermentation of soybean in the temp. Sorry, TEMPH, temp, in temp. So these are the popular fermented non-dairy food product. And idli and dosa, you know that we all know it very well. The idli and dosa very popular in South India and whole India now it is popular. And idli or dosa, idli, in case of idli, generally rice and rice and black gram mungo or urd beans are generally used for the production of idli. And here the Leuconostoc mesenteroides bacteria are used generally fermenting the idli. And dosa, in case of dosa, also the rice and black gram mungo or urd beans are used for the production of the dosa. And it is also fermented by this type of Leuconostoc mesenteroid Pediococcus cerevisiae type of bacteria. And dhokla is here dhokla is in case of dhokla rice is used also, but in case of dhokla the Bengal gram dal is used in place of black gram dal, which is used in the idli and dosa, in case of idli and dosa. So you are seeing the so many popular dairy and non-dairy food products are available in our market, in our daily life. So we should aware about that. Now one thing I should tell about that this type of fermented products are by two way they can be produced. One is the natural fermentation or spontaneous fermentation, another is the controlled fermentation. One is the natural fermentation or spontaneous fermentation, another is the controlled fermentation. In some of the fermented product, the natural fermentation is used, generally in case of idli, dosa, and the sauerkraut, pickles, and the production of dhokla, and sometimes dahi. Also, natural fermentation is happened, because here we are not adding any starter culture from outside. It is this bacteria are generally inherently present in the material, raw material. So, and we have no control over the bacteria starter culture here, and also the affecting environment like temperature also. This is called natural fermentation. And another is the control fermentation, where we are using the multi-strain starter culture in the form of devious culture for the production of yogurt, for the production of other fermented dahi also. We are producing this type of for multiple strain starter culture, defined starter culture for the production of, and we have the control over the starter culture. Generally, this before fermentation, this product, this milk are heated at high temperature to destroy the all the inherently present microorganism there, and freshly added the starter culture there to control the whole fermentation process very well. So we can get uniform product from this controlled fermentation. Here also the incubation temperature is also controlled very delicately. So this product is very uniform. There is no problem. Same product we will get in every process. So these are the two types of natural fermentation and controlled fermentation. Now we are talking about the effect of bacteriophage on the starter culture. We know that bacteriophage are killing the bacteria, bacteriophage or virus are killing the bacterial cell in by lytic cycle. We know that. Now, in case of this, in this case, the it is a when we are adding the starter culture, any devious culture, direct vat set culture in the milk or any food product for the production of any fermented milk product. If there is a fermentation is not happened there, if the bacteria present in the starter culture will be killed by this bacteriophage attack, then there will be very large amount of economic loss for the industry also, and it is a very hazardous process. If a huge amount of quantity of milk will not be fermented properly, then it will be very problematic. And in dairy industry also, in the food industry also, these industries are facing every day this type of problem, with the attack of bacteriophage on the starter culture and stop the fermentation subsequently. So this is the problem of the day. Problem of the day. We have to be careful about that. How we can prevent this type of bacteriophage attack on the starter culture. We know that bacteriophage are killing the bacterial cell, means starter culture. Now, generally, the sources of starter culture, the sources of bacteriophage in dairy industry. Bacteriophage in dairy industry. First one is the uh, whey. Second one is the effluent. Third one is the air. Fourth one is the unsanitized equipments or pipelines. Equipments and pipelines. Generally, whey is a very good source of virus. Virus are generally growing, bacteriophage or virus are generally growing at pH 6.5, optimum pH for their growth, and at 30 degrees centigrade temperature. And we know that to kill the virus particle, it is very difficult job. And whey is the main source of virus, because the whey will contain, means whey is generated from the production of paneer, where the paneer will be produced, or cheese plant. There are huge amount of whey is produced, and this whey is a potential source of virus or bacteriophage, because the bacteriophage requires for their absorption method to attach with the bacterial cell for their attack the bacterial cell. They need the calcium, like so many minerals. So whey is generally, we know that whey is generally rich in, say, this type of minerals, calcium, potassium, sodium, like that. So these are helping the virus to attack the bacteriophage. The whey is the good source of, good medium for the growth of virus or bacteriophage. So we should be careful about that. So whey should be disposed very away of the plant, so that it will not contaminate the starter culture, main starter culture. Effluent, you know that if in effluent ETP plant, the effluent will be very there are so many bacteria and is present, and also virus will be present. There will be no control there. There are airborne, and they, from there they can contaminate the starter culture also. They can attack the starter culture. Effluent and airborne, mainly virus are mainly airborne from this effluent, and where they come through this air to attack the bacterial cell or starter culture. And unsanitized equipments and pipelines are also the source of, very good source of virus. Virus particle can survive 1 to 10 years in the equipments or pipelines as it is. Means they can survive 1 to 10 years. So they can survive long time if they grow there. And sometimes what happened, on the pressure of work, for the pressure of production, generally production people are skipping the sanitization, cleaning and sanitization process sometimes, for the to produce more, due to the pressure of the production, to produce more. So this should not be happen. You should in every time you should clean and sanitize the equipments and pipelines properly, so that there will be no chance of bacteriophage or virus attack. So these are the main sources of bacteriophage in dairy industry. We are talking about mainly the dairy industry. Mainly starter culture are mainly used in the dairy industry and the breweries industry mainly. And breweries industry generally yeast are used, but in dairy industry bacterial starter culture are used mainly. So, and now how to the uh, process prevention of bacteriophage attack, prevention of bacteriophage attack on starter culture. What are the methods? Attack on starter cultures. Heating any medium or any equipments or any for sanitization purpose, heating it at 70 to 75 degrees centigrade for at least 30 minutes generally kill the virus particle or bacteriophage particle. Heating at 70 to 75 degrees centigrade. And then is the chlorination of whey chlorination of whey at 600 ppm generally kill the all the bacteriophage present in the whey. Third one is the sanitization sanitization with paracetic acid, sanitization of equipments or pipelines, paracetic acid at 200 ppm concentration will kill the bacteriophage particle or virus particle. And fourth one is the rotation of culture. We know the bacteriophage and virus are very strain specific. So we have to arrange and store the different strain of the same bacteria starter culture with us, and we have to rotate rotate it in the course of 2 weeks or 14 days. We can change the bacterial strain or starter culture so that the virus will not affect it properly. Virus is not attack the culture. Rotation of culture is important, and use of multiple strain culture. Use of multiple strain culture. I have already discussed what is multiple strain culture, which are generally fudge resistant, and sometimes for this starter culture, one strain will be attacked by the starter culture, but the other strain will continue the fermentation process or acid production, but the acid production will be not not will be in the desired quantity. So this will be problem for that. Sometimes what happen, we see that our fermentation, where the fermentation will take at 40 degrees centigrade for 8 to 9 hours, it will take 16-20 hours, but the acidity will not reach to this amount, to this quantity. Like for the buttermilk preparation, when we are incubate the milk after adding the starter culture, generally it at 40 degrees centigrade, it will take 8 to 9 hours to reach the point 0.9 acidity of lactic acid. In terms of lactic acid acidity 0.9%, but sometimes we see that after 8 hours it will reach only 0.5%. So what happen in this case, it will the fermentation will not be stopped entirely, because one bacteriophage will attack one strain only, but another strain will continue to grow. So they will grow and produce the acid, but not to the desired quantity. So this is the problem sometimes we are facing in the industry, in the dairy industry, particularly for the production of buttermilk. Practically this type of fermentation in production of dahi also, we can see that desirable body and texture and the starter culture in the prescribed time, it will not get the fermentation properly. So these are the main problems of that. So this is the solution of this problem, multiple strain culture, but it is not a total solution of the problem. Also there will be some your time will be wasted, and the proper product will not be developed. Will not get the desired body and texture of the product by this process. So rotation of culture, one of the solution of that. And sixth one is the attenuation of fudge. Attenuation of fudge means, attenuation of fudge can be like that we are producing the vaccine, attenuated culture of vaccine. Like that we can attenuate the fudge by by going through the some cell culturing process for long time, we can attenuate this culture. This is another thing to check the growth of the virus. Attenuation means, attenuated culture means it will be viable for vaccine production, but it will lose its virulence factor, means disease producing factor it will lose in case of bacteria and in case of also virus, the attenuation of phage. And seventh one is the seventh is the production of phage resistant production of seventh one is the production of phage resistant strain. Phage resistant strain starter culture. How we can achieve it? By the we can grow the starter culture in presence of some phage. What happens? Sometimes phage will go to the lysogenic cycle, and they will integrate their DNA into the starter culture DNA. So that's why the starter culture will acquire the resistant power to this particular phage. This is called super infection immunity. By this way, we can develop some phage resistant strain like that. And another is the development of, generally in the debris culture, this type of uh, starter culture is it development of phage resistant starter culture, phage resistant starter culture by genetic manipulation. Means genetic recombination process, we can integrate the phage resistant property in the manipulation. We can integrate the phage resistant property gene in the starter culture, so that they will they can protect their self from the phage attack. So in this way we can prevent the bacteriophage attack on starter culture to curb the huge economical loss and production failure, and on the hazards also, because this due to the starter failure by the phage attack, the whole product, huge amount of product will be wasted, and it is very difficult. It will create the environmental problem also if you dispose all these things, and it is